In the previous activity, you were able to experience counting cells using a counting chamber known as hemocytometer. This is just one of the methods used to count cells. We will discuss briefly the four general approaches used for estimating the number of unicellular organisms. It is also often essential to determine the number of microorganisms in a given sample. For example, the ability to determine the safety of many foods and drugs depends on knowing the levels of microorganisms in those products. A variety of methods has been developed for the enumeration of microbes. For unicellular microorganisms, such as bacteria, the reproduction of the cell reproduces the entire organism. Therefore, microbial growth is essentially synonymous with microbial reproduction. To determine rates of microbial growth and death, it is necessary to enumerate microorganisms, that is, to determine their numbers. The first method is direct count of cells. Cells are counted directly under the microscope or by an electronic particle counter. Direct microscopic counts are performed by spreading a measured volume of sample over a known area of a slide. Specially constructed counting chambers such as the hemocytometer simplify the direct counting procedure because they are made with depressions in which a known volume overlies an area that is ruled into squares. Fluorescent dyes are becoming more used in recent years for a variety of procedures, one of which is bacterial counts. These dyes can be employed to stain all species, a particular species of interest in an environmental sample, or even a specific component of cells. The second method is indirect count of cells. Microorganisms in a sample are diluted or concentrated and grown on a suitable medium. The development of growing microorganisms is then used to estimate the numbers of microorganisms in the original sample. The most common procedure for the indirect count of bacteria is the viable plate count. In this method, serial dilutions of a sample containing viable microorganisms are plated onto a suitable growth medium. The plates are then incubated under conditions that permit microbial reproduction so that colonies develop that can be seen without the aid of a microscope. It is assumed that each bacterial colony arises from an individual cell that has undergone a cell division. Therefore, by counting the number of colonies and accounting for the dilution factor, the number of bacteria in the original sample can be determined. Another type of indirect count is the MPN, or most probable number. The MPN procedure is a statistical method based upon the probability theory. Samples are serially diluted to the point of extinction, that is, to a point where there are no more viable microorganisms. The pattern of positive tests or growth in the replicates and statistical probability tables are used to determine the concentration, the most probable number of bacteria in the original sample. The third method is direct measurement of microbial biomass. Cell mass is determined directly by weighing whole cells Biomass can be correlated with cell numbers by reference to a standard curve. Wet weight or dry weight of bacteria may be used for estimation of cell numbers. The last method is indirect measurement of microbial biomass. Microbial biomass is estimated by measuring relatively constant biochemical components of microbial cells, such as protein, ATP, lipopolysaccharides, peptidoglycan, and chlorophyll. Biomass can also be indirectly estimated by measured turbidity that can be correlated with cell numbers by reference to a standard curve. Again, monitoring the total bacteria count is necessary because the number of microorganisms shouldn't exceed certain guide values.